I'll share my screen. Can you hear me nice and sound? Good evening, ladies and gentlemen. I'm going to present my proposal for this genetic engineering project. It's called Engineering a Photostable Wild-Type Green Fluorescent Protein Variant via serine 65 to NQ69 to methionine mutations. So the importance of this protein is that it was revolutionary in the field of genetic engineering. It transformed cell and molecular biology by enabling the visualization of proteins in living systems, a thing that was thought not to be possible before, but with this protein from this jellyfish allowed us to make this possible. So this protein has the ability to form a fluorophore without adding anything extra, and this allows us to track protein dynamics, visualizing gene expression in vivo, and also to start visualizing how things arrange themselves in space. One thing is to know something is there, but the other thing is to actually start seeing where the proteins are located in a system. So the original wild type, it's called AB GFP. It's still the basic protein for many of the different variants that exist for this fluorescent protein. However, there is always something that you need to sacrifice when you make a new variant. Sometimes it enhances luminization, but it still has photobleaching. In fact, photobleaching is one of the things that many of them are still suffering. And this refers to the process of irreversible loss of fluorescence upon prolonged illumination, because the bright side breaks. So that happens. So this usually restricts the ability of scientists to perform long-term experiments that can be useful, and also sometimes doesn't allow them to visualize so effectively when a protein is actually being activated, right, or excited. There are many different variants again, and there are different sites reported that had been engineered. So it's a thing about the reason that there are many different variants is that you kinda tailor your protein to your needs. Maybe you don't need a protein that is too stable, but need it more bright. So it depends. In this case, I want to focus on photobleaching. So it's crucial to keep developing new GFPs, to keep pushing the boundaries of biomedical research, to make studies, long-term studies of metabolic processes, to actually capture these physiological processes in vivo, which are kind of fleeting. So engineering, in my research, I kind of observed that it was necessary to still make improvements on these proteins. Although, well, I think to mention here is that this is not the only source now of green fluorescent proteins, but it's the base for our understanding on them. So what's the structural properties of this wild type green fluorescent protein? It is an 11-stranded beta barrel, like a cylinder, as you can see in this image from pymol that I was able to generate, although not easily. Inside the barrel you have the core, and there is a short alpha helix that threads through, which is this on the lower image. That alpha helix hosts the 3 amino acids that undergo the catalytic reaction for the brightness, which is CER65, tyrosine 66, and glycine 67. So this is the center that absorbs the blue light, the UV or blue light, and emits the green light, although some variants also emit orange or red light. What's interesting about the wild type is that, again, it doesn't really need to have something added to the catalytic site to perform the reactive the reaction that provides the fluorescence. So as for the structural properties from protein data bank, this is the ID of the wild type protein, the ABGFP is 1 GFL, and well, this is the one that I use. It took me a long to find it, because since you have a lot of different proteins stored, then for a moment I thought that that one was the wild type, and it turned out that it wasn't, because it already had some variation in one amino acid, and then I had to go back and search on the internet. It was quite difficult. This is the official protein reference from UniProt for the wild type, the P 42212, and these are the amino acids. There's a typical start for this protein, which is the MSKG. There is another that has a V, but that typical start is for another type of green fluorescent protein. But the one from this jellyfish is this one, MSKG. Also, I got the DNA template to because I wanted to modify, or I wanted to make sure that the databases agreed, like the template and the protein, the UniProt sequence agreed. So first I had to get the succession, which is the M6265-3, and get the open reading frame. So the open reading frame starts at position 26 and ends at position 742, which are the ones that you can see underlined and highlighted in yellow. So when you translate this into its protein sequence, it's the same protein that is stored in UniProt. It's the same characteristic start, the MSKG, and the typical 238 amino acids, which is exactly the same. So now, at that moment, I was sure that I had the right protein. So a comparison with an existing protein, engineered protein, which is the enhanced GFP. Well, the hallmarks of the two mutations of the enhanced GFP are the F64 to lysine and the serine 65 to tyrosine. So that's why it's called enhanced, because the leucine 64 improves folding and at 37 degrees, and the tyrosine 65 increases brightness. So it is more robust than the wild type, but still has issues. I choose to showcase this one because it's one that scientists use a lot, the enhanced one. So it still has the issue with bleaching and blinking. So that is why new proteins were developed later on. But yeah, you can see the difference, for example, in the F64 and the change to leucine 64. So you can notice that you have a ring there, and in the other amino acid you don't have that. Again, these are the other differences: the serine 65 for the tyrosine 65 in the enhanced one. The glycine, well, it stays the same. It only looks a bit different. I think it's because of the files for the pymol, but it's supposed to be the exact same amino acid. So the engineering proposal was to combat photo bleaching through changing some amino acids. So the engineering is to modify serine 65 for tyrosine 65 to shift the excitation maximum from 1395 to 4088, and the Q69 to methionine to improve the photostability to combat the glutamine, for methionine to combat the photo bleaching. So this creates a tighter cage because of you create an hydrophobic shield. So this is what I found reported in literature. The thing is that I didn't find like this combination, so I thought it was a nice thing to propose it as something new. in simple terms, I just choose 2 different amino acids that could give me this effect. So the thing was the molecular strategy was just to make the changes with mutations, so changing the codons so that you had the different amino acid changes. So here it will result in these red ones, and these are the positions of the amino acids to be modified. So then site-directed mutagenesis, I choose like this plasmid because it will be easier. So you could make direct site mutations. As you can see in this diagram, you have your plasmid, but you also have the mutagenic primers with the specific mutation, and you perform it 2 times, and then you have the protein, as you can see here. You can see here, like, the first mutation is the serine 65 to tyrosine, and the second round performs the 69 mutation, and then you have your product with the 2 mutations. So then as for cloning, this is just a cloning vector to have a quick mutation for a quick experiment, because you could tailor to have it in mammalian cells, but I thought, like, just have the cell first and then maybe choose another animal or another vector. So this is in E. coli with this vector PET28A, and finally, well, that perform a validation assay, a bioluminescent assay to see which one decays faster. So lastly, I just perform a structural prediction in this AlphaFold 2. I just charged my modified amino acid sequence, and it gave me this new protein, which I also loaded in pymol. I made an alignment with the wild type, and as you can see here, it's almost similar, almost the same, except for the mutated sites, which are the Q69 and the 69mm, and also the serine 65 for the new GFP for T65, and they look different. So that would be it. The thing here is that, well, you have to try things, but I think that starting from the wild type to make simple mutations could give the protein, well, more stability. That was my thought. Thank you.
Your presentation was very good. You just need to practice the time.
Yes, I got.
What you are interested in that? In what cases this auto bleaching affects the function as a biosensor of this protein? What is the problem related or linked with this auto bleaching? Because it was not clear for me what you want to improve the molecule, taking out or avoiding the auto bleaching.
Okay, well, personally, it was just because I thought it was better for me to understand the genetic engineering process with this protein, but also I always like medical sciences, so sometimes we still lack some tools to perform in vivo experiments, which could be very helpful for cancer studies, to study metabolic processes, maybe things like that. Maybe we have advanced techniques, but to make it more available, that was like my reasoning.
But in your research, did you realize that the auto bleaching is a limited step in the applications of this protein, or in what cases it is a problem, the auto bleaching?
For more than anything, for long-term experiments, if you needed the protein to be functional for more than a few minutes, maybe some hours. So there, okay, it's where when I found that it could be an opportunity, and also to make it. I think it's cheaper when you standardize a protein, because if you have to modify it yourself for your own experiment, so the more variety we have of these proteins, I think the better.
Okay. Yes. Okay. Perfect. Perfect. Thank you so much, Alberto. So claps for the answers of Alberto.
Thank you, doctor.
Very good. So thank you. Let's go to Alejandra Treviño, please.